So 11 o'clock and welcome to another Chai Advice. In Mumbai, it's raining very heavily since last 3-4 days. But thank you for coming on a Sunday. And with us today we have Lavia, Padmalaya, and Javi. Speakers, please come. Please come. So today's speakers are research scholars in the Department of Biological Sciences at TIFR. And today they are going to discuss with us something very fascinating. And that is like, see, we all are used to seeing something big, macroscopic, that we can see with our eyes. But how do things develop from a small microscopic cell to a big macroscopic object like us? Or let us say other way around, like how can we imagine what is happening from big thing to a small thing? And to tell us about that and the fascinating science they do at their lab, they are here and they will take, I think they will, you take turns in explaining. Okay. So every one of them is going to tell us small part of what they do. So let's, okay. So, Oliver.
I will start.
Now you can start with the question.
I've done that. Okay. So, good morning, one and all. I am Lamya. Three of my lab mates who are Padma, Charvi, and Anupriya. And what we are going to be doing today is talk to you about how an organism takes its shape. Particularly, is building an organism something similar to building a house. So that's the analogy that we're going to be building on. So here we go. We are from the Department of Biological Sciences at TIFR. And yeah, we're going to talk about what really piques our interest and something that drives us, even though we look at the same process every single day for the last 3, 4, and 5 years. Okay, so here we go. So actually, just to get you a little excited about, actually looking at organisms and what I mean by building an organism. So let's see one such organism and actually just look at a movie where the embryonic development of this organism happens. And this organism I'm talking about is the fruit fly. And it's one of our favorite organisms because this is the organism we work with in the lab. We breed these flies and we work on their embryos. So it's called the Drosophila. It's actually the fruit fly, and I'm sure all of you are aware of it because you might find them annoying and hovering over ripe fruits and bananas, but they are very dear to us. So saying any of that would offend us. With that, so I will just play this video, and so this is the fertilized egg of this fruit fly. And if we are building an organism, let's just see as a view, as a video, what exactly does this mean. So looking at an egg, and wow, there are these movements which are happening of individual cells that you can see are marked. There was a certain structure that went up to the left side of the embryo, which is actually going to form the head, whereas the right side is going to form the tail of the fly, tail end of the fly. And now we see that there's slowly some kind of backward movement, something that we call as axis straightening, because the fly is not going to be curled up on itself, it's going to be a straight organism. And here you see some tissue which is actually not a part of the adult organism, so it's just going to shut up. So you see two intervening sacs which are going to come close to each other and shut the gap, because at the end, the organism cannot have a hole in its adult structure. So, and you see there's some highlighted bit up here, which actually is the nervous system of the fly. And that is something that I work on as my PhD project, is to actually understand how do we take each cell and make an organ like that. So that is what my PhD is about. And you will see some twitching movements at this point, because this is when the muscles have been formed. And so now the embryo is showing some kind of muscle contraction, which is why it's showing the kind of twitch. So this is great, and I hope I can get you some interest into like what we actually do, because This is one of my most favorite movies where we see actually a 17-hour process in like a time lapse of like 2 minutes. Okay, so moving on. What am I going to talk about is to actually understand that when you start with a single cell, it's going to divide, proliferate into a group of cells, all of which are completely homogeneous. They all look identical to each other. So at what point does the symmetry break? What point? And to do so, you need a plan, which is what the blueprint is, something that's the blueprint of the house. Basically, to understand that where do you form the kitchen, the living room, the bathroom, and so forth. So we are starting with just a simple organism, and now we are going to make segments into it and establish some kind of polarity such that it decides which end is going to become the head and which end is going to become the tail. So that is the kind of flow we'll try to follow. That is, we start with the blueprint. Next, Padma is going to talk about then how the individual rooms get their functions. And finally, Charlie will talk about that to make these rooms, what are the kind of materials that you require, the kind of cement and mortar and the building that requires, sculpting that requires to make a functional bedroom, let's say. Now to understand that basically now if these cells are not specialized at the right time, at the right position, it's going to be a haphazard mess, which means that either some cells will get their fate and will become an organ at a misplaced position, or they might not form an organ at all. And also because if it is sometimes that for the organism, it's extremely illegal that all the cells integrate into the body plan of the organism. So that's why what we have, what's absolutely essential is that there is a regulation in the patterning, what we call as the patterning of the body plan of the organism. And also I just want to get your attention towards that basically this polarity establishment. We're talking about an embryo, so just imagine the scale at which we've come down to. We've come to a single cell which has formed an embryo. And at this point, this is the crucial developmental plasticity window, which is when things, if they go wrong, it's going to mess up the whole body plan. So at this point, it decides the future 40 years of your life. So I hope like you get the enormity of the situation that we are talking about, and that's why it's one of the most highly controlled and regulated times of a development of organisms development. So now when I was talking about how do we set up a body plan of an organism, so we start with segmentation. By segmentation, I talk about basically that what forms the head and what forms the tail end, which means that what forms the anterior and the posterior axis of an organism, which is similar, and this kind of segmentation you see in us as well. Like for example, our vertebral column, it has been segmented into the cervical, thoracic, sacral, lumbar, sacral, and the tail end. Just like that, we have our organism, which shows this kind of segmented pattern. So you will see that it shows these superficial segmentation pattern, which translates into the adult. That this embryo translates into the adult, and you see that there is a head region, there's a thorax region, and the abdominal parts of it. So this, yeah. So just like that, the fly will also have an anterior to posterior axis. Okay, this one. And the segment that I was talking about here is something that we saw in the movie here, which are these segments within the embryo, which actually help a cell within an embryo to get some kind of positional information. It's like it's like saying that what is my coordinate within this embryo, just like latitude and longitude help you to specify what your location is. Just like that, in an embryo, these segments help them to tell them, okay, where do I lie with this entire axis? Okay, so now all of that, so you get the point of what I'm trying to say, that why is it necessary to build an anterior-posterior axis? So now we begin to actually building that. How do we come to that, and how do we determine what are the signaling mechanisms which decide what forms the anterior to the posterior? And this is work which has been started from work during in the 1960s and the 1970s. and where it has been very well established that it's a morphogen gradient mechanism that helps to specify this embryo possible. So what do I mean by morphogen gradient? So morphogens are just signaling molecules. They're signaling molecules which are released from one cell in a concentration. It's released from one cell, and the concentration will change in a distance-dependent manner, which means that the releasing cell will have higher molecules versus the cells which are around it. Because of the distance, it's going to diffuse over space. Now, what is the smart thing that we have is that each of these cells is able to respond to the molecule in this concentration dependent manner. Such cells which are basically exposed to higher amounts of this morphogen will become phase 1 as compared to another molecule which will become like as compared to the far end of it which will become phase 3. So I'm just going to do a simple
So please stop the screen share. We can.
Okay, so I'm just going to move towards showing you what exactly do I mean by morphogen gradient. So here, what we are, it's a very simple test that I'm just trying to develop some this. So I'm just going to take a blue color band, which is basically yeah, that's it. So basically, you have something which is just going to mix it. So let's say this is a group of cells which are going to be releasing a particular morphogen. And this morphogen, I'm going to do a series of serial dilutions, which is basically this morphogen is now going to spread over the entire distance of the embryo. So as you will see that the color of this gets fainter and fainter as you move away from the source. Now, let's add to this complex, which means that the cells which are closer towards the source are going to take up, let's say, the head, the identity of the head, whereas towards the tail end, it will take up, as in towards the other end, the posterior end, it will take up a more tail-like morphology.
No.
Now, I will just increase the complexity by a bit and see how this becomes more refined. I'm going to add something which gives yellow, red color. I'm just going to, let's say, mix it up a bit. And now I'm going to give another gradient which starts towards, which is higher at the other end of it, such that it will get now, again, we do some kind of serial dilutions and time. And what you see here is actually a spectrum of colors. And so you can tell that the ones which have higher amount of blue is going to become more head-like, whereas those in the more at the other end, which are more red color, or the morphogens are more of a different nature, are going to become the tail end. Whereas those intervening concentrations, like this would have, let's say, X concentration of molecule red and Y concentration of molecule blue, is going to pull up, let's say, the abdominal identity to this region, whereas these others are going to form, let's say, the mouth parts. So this is the way we can have really fine gradations, and the cells can form these segments and can take up.
Just for the benefit of online viewers, can we just do this thing in front of camera again, because I think we missed focusing on it for our technological constraints. So if you could bring it this side, just bring the beakers this side.
Yes.
ऐसे रखिए, so I can go ahead.
So fine, what I have done here is I have done a series of dilutions here, wherein we started with adding a more concentrated. So these are a group of cells which are going to be releasing certain morphogens, which are basically signaling molecules. And these molecules over time are going to spread in the entire embryo.
Okay.
But
so is it like you are starting with different kinds of cells at different locations?
Cells in different locations. So let's say this is an embryo out here that we have around, and this is the far, let's say, the head end of the embryo. Can't really say the head end because this is the event which is determining the head end. But let's say this is one pole of the embryo and this is the other pole of the embryo. And those which are expressing more amount of blue are going to become the head, whereas those expressing more amount of red are going to become the tail end of it. And as we go and as these molecules spread towards the central part of the embryo, we'll have more finer gradations and regulation, such that one group which is expressing a particular cocktail of, let's say, blue and red is going to express a particular identity of cells. Let's say it will become the trunk part of the embryo, and the other part, and the other which has more amount of blue, or rather like very less amount of both these morphogens, is going to become, let's say, the chest region or the thoracic region of the
So is it like kind of a democratic process where more cells form something predominantly what it is intended to be?
I wouldn't say so at this play by democratic. Okay, so we'll say that, and then actually we'll come to that in the next segment of it, wherein we talk as to what gives this cell the power to release these kinds of morphogens. And I'll go back to the screen and all right. So okay, now that we've gotten well versed with at least what morphogens are,
Yes. Click on those three dots. Hide. Oh, okay, fine. I think that's gone. All right, we are good to.
I see that I am unable to change screen.
No, your screen is shared.
I know, but it's unable to change it as of now.
Oh, okay.
I think my
Frozen?
laptop, which is an everyday problem.
Oh, okay.
Okay. Okay, cool. That is good. All right. Okay, fine. So now I am going to come back to our mighty slide. And I'm going to tell you that how exactly do we form these gradients. So now it's just that we take an embryo, which like we saw in the first slide, which was like a homogeneous number of cells. So now the mother, that is the parent fly of this embryo, is going to deposit certain molecules at one end of the embryo, and these molecules are now going to diffuse towards the other pole. And after this occurs, and that is the gradation that you see. So this is something that we call as a biomass, where you see the colors which are red, yellow, and white show higher amounts of higher concentration, whereas those diffuse towards the other. And now we see that as this gradation occurs, what this translates into is that these cells now express, let's say, the gene hunchback. Names don't really matter. But and the other end, which has a lower concentration of this, express insulin. I just want to tell you that these are not some pseudo-colored images. These are actually images which are acquired with a microscope. And what we have here are probes which are fluorescently tagged against these proteins. So these proteins are only found in the head region, which helps to specify the head identity of the embryo. Whereas pupil and all will help to identify the trunk region of the embryo. So like that, and this is one set of transcription, or rather one set of proteins that I am talking about. There are a plethora of these, and this gradation, or rather this segmentation, becomes finer and finer as more morphogens are added to this entire to the system. So with that, so it's all fun. It's really nice to learn about how this happens. But the painstaking work that goes on to find out a result like that is something that I absolutely enjoy, and I think history cannot be left out of some of these seminal work that has been done. So this was one of the groundbreaking research which came from US, Matthew Wolfhard and Eric Weisstrom. And these guys won the Nobel in 1995 for their invention, or rather not invention, the discovery of these segmentation genes, that what tells the embryo that you are the head end and you are the tail. So this is the two of them in Princeton, and they would actually, so they were just a two people run laboratory, wherein they would sit with this microscope which has like two eyepieces, and both of them would screen larvae after larvae, embryo after embryo, looking for defects. So I'll go a little bit into like how exactly did they do that. So how do I say, like, that there are these segmentation defects? So basically this is the larva stage of this fruit fly embryo. So once the embryo has reached the end of embryogenesis, it hatches into a larvae. And we have some of the flies which have these larvae on the wires, and we shall show you at the end of the session. And you will see from the larvae there are these alternate stripes of hair and cuticle, hair and cuticle. And this is similar, and this is just, it kind of represents the segmentation that you saw in the embryo. So that kind of mimics that. So it arises because of that. So this number and their position are fixed in a wild type scenario. Now, these two, they did a mutagenesis screen, which is basically, they took these embryos and they exposed it to a number of mutagens. So now, because of the mutagenesis, which is happening randomly in all the genes of the fruit fly embryo, what you see are there are defects which occur in the segmentation pattern of them. So now you see So there have been, and also what the fun part of about lives is that the way the genes have been named is on the basis of the mutations that you see. So there are genes which are called as even skipped, which means that all the even number stripes have been skipped, and you only see stripe number 1, 3, 5, 7, and so on. Or there are odd skipped genes and so on. So here is how they had actually seen. So they discovered 15 such genes which are involved in patterning of the fruit fly embryo and giving rise to these determining its body plan. And finally, the work of Edward Lewis, what he did was this is something called as the Hobbs genes or the homeobox genes. So now we've understood that, okay, we have broadly gotten it that, okay, this is the head end, the trunk end, the tail end of it. But now how do I actually say, okay, in the head, we need to have, let's say, the head structures which are there. Then there are in the thorax region, like you have the T1, T3, the thorax region, and so on, the abdominal segments. So to get this finer identity of these segments, what we have is called as the homeobox segment genes. So what these genes are basically, and that it's so well orchestrated and so well defined. In the sense you have these set of genes which are present on the chromosome, and they are present in the exact order in which they are expressed in the organism. Which means that the pink gene here will be expressed in the head, the orange gene here will be expressed in the thoracic region, whereas the green will be expressed in the tail end. So they are present in that sequence only, such that when we did mutations, wherein let's say so this is called an antinepedia, name again doesn't matter. So we take this and let's say we mutate it such that we remove this and add it to another location. So what we saw was the antenapedia, which is the orange region which forms the legs or the appendage, actually leads to the formation of legs on the head. So these are called as homeotic transformations, and that is what blew a lot of people's mind because they saw these mutations come up. And like that, you can play around with any of these genes and put them anywhere, and another body part will come out from them. And this is not something that is only true in flies. This has been done in amphibians as well, wherein they would transplant certain cells from, let's say, the leg and put them in the head. And this is true in humans as well. Of course, this is, but just that the number of genes are much more in humans as compared to in flies. So with that, I will move on and we shall ask Padma to like take over from here.
Online.
Sure.
It means that we have a reasonable handle on how many legs, for example, an organism can grow. Like we can grow as many as possible, we can chop them off. Is it possible?
Yeah, you can totally have, but again, it has to be in a particular, so if you're saying that can we have more legs, that's exactly what happened here. So if you duplicate a gene and add it at a different locus, then it's going to get expressed in that locus. So let's say I take my genes which are present in the legs and put it in that, since I told you that it was a sequence in which the genes were arranged. If I remove this and add it to any other, that gene is going to get formed there as well. So yeah, it's extremely million.
There is no way that they can come back.
No. So like so that brings us to a very interesting thing of called as transposons. We have something called as these viruses which are able to cut genes from the middle and place them in random locations. So this happens in all of us. So they're all jumping genes for that matter. But the probability of that happening is like really high. I mean, of it happening is really low, as in the sense that it will. You understand that we have 23,000 genes, and for that to be able to remove at the precise location and add it to another location is quite unlikely. And also, we have also a lot of error and DNA. What do you call it? Repair mechanisms, which will not, like you have to understand, these are very robust processes. Evolution has really shaped them over a very long period of time, so it's not possible to mess it up with anything.
I think the person is answered at it.
All right, okay, cool. All right, so then we'll move on to Padma. She's gonna now tell us that fine, we know where things are formed, but how exactly do you make them? Like, how do we make these forms? All right, thank you.
Speak loud.
Okay, so now that you all have a, okay,
So now that you all have a fair bit of understanding that the embryo has gotten its blueprint, so the embryo now knows where it has to form a head and where it has to form a tail or where it has to form its abdomen. But exactly how does these different kind of cells, they change their shape to actually form different organs? So before I dive deep into it, I'll just like to include or say this famous quote by Louis Wolford, a very famous developmental biologist, who said that it's not birth, marriage, or death, but gastulation is truly the most important time of the life. Why? Because as you can see, and you all are familiar with this particular movie, this is a dorsal embryo. You can see the top is we are viewing the embryo from the top. The bottom is we are viewing the embryo from the bottom. Now, this embryo is basically a single layered embryo. So, this is a tube. This is single layer. Now, from this single layer, if I play the movie, you can see that these different cells, basically these dots, take it as different cells. These cells are moving in a very coordinated fashion. Let's play the movie again. What you can see that this movement is not very random. You can see there is a certain group of cells which will be moving backwards. Yeah, they will be moving backwards. And then after some time, you will see some group of cells that are coming in a coordinate or in a particular direction. So all of these movements are very, very choreographed. But how does it actually happen? Now, as I said, the embryo is basically single layered cells. Now, all of the organs that are there in our body, they are basically derived from three different layers of the cells. So from this single layer embryo, I have to make three different layers. And what are those layers are basically, first is the ectoderm, which is fairly simple, the outermost covering of the body. So if you consider this as building a house, the ectoderm would be similar to the brick or the walls of a building, which protect us from the external environment. And here, similar to this, the ectoderm is basically help us to protect us from the external environment. Yeah. Now, from that, the mesoderm, which is basically the middle layer, and this mesoderm would be one layer beneath this in an early embryo. Now, mesoderm, the cells which we call mesoderm, they basically form muscles, bones, or circulatory system in our body. So imagine a building without these rods. The building will collapse. Why? Because these rods are basically they provide the framework in a particular building. So muscles, similar to this, muscles and bones, they provide structure and framework to our body. Now coming to the circulatory system, the basic function of circulatory system is to transport oxygen and gases to the entire body. Now if we consider this to our house, in our house the major energy supply is obviously the electricity. So the mesoderm is like a middle layer of the cells which give rise to muscles, bones, and circulatory system. Now comes the endoderm, which is the innermost layer of our developing embryo, which is similar to the plumbing system. So in our what is the major function of plumbing system in our home? It's basically the removal of or the processing of waste and removal of waste, basically. So in our body, the digestive and the respiratory systems are the ones which take out all the process our nutrients and take out all the nutrients. Now, from these different layers, the ectoderm is pretty simple because it is the outermost covering. It is set already. Now, the problem comes how do we position the mesoderm and endoderm, which has to be there inside the embryo, because the muscles, bones, or the digestive and respiratory system, they have to be there inside the embryo to have to be protected from the external environment. So we have to make a different layer which is there inside the embryo. So first we talk about the positioning of mesoderm. So from this single layered cells, how can we make a second layer of cells which is there inside of this embryo? Okay, I'm using this paper again and again. So if this is a circular cylindrical thing, I'm pressing it here like this. Hold it. Yeah. I'm pressing it here like this. So it will first form a furrow kind of thing here if I'm pressing it exactly in between this, and then it will form a small tube here, right? In the midline. So if I play this particular movie, and this is a movie of a Drosophila embryo, what I want to focus in all is to focus on this particular act. Okay, we'll continue. So I would want you to focus your attention to this particular part of the video. And what you can see, I'll play it again. And what you can see that these cells which are there on the outer layer of the embryo, they are sort of moving inside, and they are making a forward. It is similar to something like this, right? Now, if I cut this embryo, if I make a transverse cut in the embryo and look at these circular veins, what you can see that when the cells are sort of internalizing inside the embryo, they are forming a tube-like structure. But once they detach from the outer covering, they make this clump, and then they spread to form a layer of the embryo, which is the middle layer of the embryo. Now just imagine a train parking in Dadar station. It is full crowded, and if you see this in the video, what you can see, the people are coming out of the local train. I just want to ask you one simple question. If it is completely parked and the door is closed, will all the people inside the train, will they be able to move out or migrate? No. The point is they have to come out of this layer or come out of the train to move to their respective place. In this case, the cell doesn't, when the train comes out of the outer layer, it does not go here and there, it migrates and form a layer inside of the embryo, which forms the middle layer of the embryo, basically, which starts with the formation of a furrow, then it attaches and then it forms a layer inside of the embryo. Okay. This particular tube, which has sort of small tubes that form here, instead of forming this clump, what we can do, this pincher here, we can form a tube inside the tube. So what I'm saying is basically, if this is there, and I just, if it is like this, and I can pinch it here, there's a tube inside a tube. Now, in our body, we have brain and we have spinal cord. So when it actually develops inside an embryo at a very earlier stage, how does it look like? It looks like this. So this is one single layer. What you can see is that the two layers are coming and they are very densely fold. Which finally forms our spinal cord. So from having a single layer, we are not taking single cell out, we are folding it and we are pinching it to form the spinal cord, basically. And this is the same transverse view of that, forming a tube inside a tube. And now let's talk about a very fascinating organ, which is the wing, Drosophila wing or fruit fly wing. The wing cells, they develop from a very circular, so the wing develops from the kind of cells which are sort of very circular in shape. So if you look at these circular structure here, yeah, if you look at this circular structure here, this is the part from where wing develops. Now you can see that this is very flattened and very circular in nature, and there is a line here. So there, as Lamya already said, that there are different genes which pattern the embryo. Similarly, there are different genes which pattern the wing also. So, for example, there are a set of genes which are present here, and there are a set of genes which are present in this half of the disc. Here, it forms the top of the wing is a flattened structure like here. Wing. And this is the front part of the wing, this is the back part of the wing.
Let me get stronger, please get one over there. So let's look at it.
So if this is a wing, this is the circular structure is like this. This half of the embryo will form the future top part of the wing, and this part of the disc will form the future back part of the wing. Now, what I am doing is I need one person. So what I am doing from a very flat 2D structure, 2D circular structure, I am making a very elongated structure which has a back side here and a front side here. So basically, if you imagine this as a circle and dividing it like here, she is putting her finger there, and this is the wing, and this is the front side, and this is the back side of the embryo. So basically, if this half of the embryo is the front and this is the back, when I am elongating it, it eventually becomes the two layers of the wing. Screen पे the laptop की छानी है. Now you can see the same analysis. This is the front and this is the back. Like now, they will form the wing. This is actually what it happens when a wing develops. Now the question comes, the one obvious question which comes here, having a more circular structure, how can you build something which is so elongated from this circular structure, right? This is a 2D entity like this. I'm building a 3D structure, or I'm building a 2-layered structure which is much more elongated like this. Now what one can do is basically, obviously, this structure is larger as compared to that circular structure. So what we can do is to divide. So if there are 100 cells in this, we can have many more cells here. So cells can divide, but the cell has to divide in a particular way to form this elongated structure. For example, if this is a cell, if it divides like this, so its daughter cell is placed next to it. If this daughter cell divides, then again the next daughter cell is placed next to it. But if the cell divides like this, it gets elongated to this axis, basically. The division has to take place in an oriented manner. Basically, the cell has to place one by one next to each other, and then have this. Can we divide these cells next to each other? Because the daughter cells, when these daughter cells are placed next to each other. On the other hand, if the cells divide like this, the daughter cells will be placed like this is the parent, the daughter cell will be like this, like this, like this. So we should be elongated in this direction. What happens is saying that we should elongate in this direction, which means that the cell does not divide randomly. They divide in a very oriented fashion. In addition to that, another process which happens during this development that is known as cell integration. For example, imagine this 4x4 grid, this square 4x4 grid. It has 1, 2, 3, 4, 4 number of cells in vertical axis and horizontal axis. That's 4 layers. Now we can make these 2 layers. And I make these 2 layers. This 4x4 grid turns into something more rectangular as compared to being more square. So the same thing happens in wings as well, where cells, different layers of cells can mix with each other to become more elongated wings from a more circular disc. So I know this is not very complicated. To make this a little more complicated, I'll invite Charlie to explain how does exact cellular mechanism, she'll explain the cellular mechanism which actually drives all of these changes.
Okay, so Padma spoke about wings and tube formation and the kind of what needs to happen to the overall tissue, and in case of the wings, what should happen to the cells in order for it to form okay, how exactly is it built? The outer layer holds. So how does it hold? What cellular and subcellular things happen? So, so I will be discussing how to share the building block of the object. Okay, so we'll start with the first thing that she discussed, that is making a tube. So, so this is the if you cut the embryo and look at single the second bucket, you will see the single cell layer. So if you zoom it into a particular area, you see like a row of cells, right? But right now we need to make a tube out of this, and then we need to make a curved surface out of this, and then fix that region to make a tube. So So, how exactly how does this finishing happen today? I won't be covering, but if you want, we can discuss it over time. But here, how exactly does this fold happen? We can, I will try to explain. So, just to, wait, so let's say, no, I took these rectangular looking cells, which are cuboidal rather, I will just show one surface. And then let's say I want to build it into a folded label. So now, if I put the cells to be just rigid blocks or rigid rectangles, then I will have to arrange it this way. And you have a lot of spaces that are generated in between the cells because it's like you can't this particular shape is not suitable for forming this structure. And now what is here it is curved in this manner. Here is the you call it the concave and the convex curvature. So when it's curved in this manner, you see that so the outer part of this is called vertical and this is called vertical. So in this case, you see that optically there is no chance, but basically there is a chance. But in this, when it goes to this kind of curvature, optically there is a chance and basically there is no chance. So we know one thing here, that the cells here are not rigid. Okay, that is something you know, that cells here are not rigid. And the way they change that shape here and here needs to be kind of complementary in order to form this. So this is similar to why, like this is similar to like having a race track where if you take these lines here, these are overall perimeter or whatever it will be smaller than this circle. So usually when runners run, they usually are staggered so that people close to the inner region are standing farther away from the finish line, right? So like that. So now definitely we know that if cells maintain the rectangular shape, they can't follow this. They can't form this folding. So what do they do? So actually these cells will become into like in that shape of trapezium. And you have in the trapezium, it's not like you take a rectangle and then you elongate one of the edges and then you have a trapezium. And then these form these wedge-shaped cells. And the wedges are opposite oriented. Here the long in this side, here the long in this side. So this way the cells are able to accommodate. So the one difference between building a house and building an organism is this, that the building block of a house is separate and that is very rigid. Whereas in organisms, the cell is not at all rigid. It can form very columnar, like almost thin sheet of cell to something, columns to which is a thin sheet of cell to something that is columnar, which is standing like this. So okay, so how cells which are initially like they started out as a cell that became wedge shape. So what really happens? So let us say in this case the cells are going to here. So these cells, the base is here, apical is here. The way they can form the wedge shape is to decrease the apical area, that is apical constriction, or to increase the basal area, that is basal expansion. Now, because the cell has a particular volume, so if it is only doing basal expansion, it will shorten in height. And if it is only axial constricting, it will its height will increase. And so this is what it does, but how does it do it? So there are molecules. I will in the next slide I will show how they define, but there are molecules which are not active. These are like take them to be like fibers that or like how do you call it? So you can take it as something that maintains structure of the cell. So you call them a cytoskeleton. They maintain the structure of the cell. Now, when the actin is present, so the actin is inside present of the part of the cell. There are these red color molecules. Now, by red color again, I'm showing here. It's called bias. is it connects all these actin rods or so to speak, and then it compresses them. So it brings each of these filaments toward each other. So the myosin is kind of like a it's like somebody who's holding okay, if I hold two regions and then pull them close, then anything that's these two structures are bound to will come closer together, right? So I am the myosin and the structures I'm pulling are active. So that's what myosin is. But in laser expansion, usually you get rid of the actin that is there in the basal surface so that it's present. And the opposite happens in these cells where there is this is the basal side, the basal functioning by similar mechanism, that's the expansion similar mechanism. And now, so this is what so this is the myosin molecule that I'm talking about. It kind of so the actin is bound to like the membrane of the cell, and the myosin is bound to the actin. So when myosin pulls the actin together, the membrane of the cell comes over to it. So you can imagine that what will happen, that is membrane, and you're pulling it closer to a smaller area. So it actually forms ruffles. You can see ruffles within the cells which are extracellular. And this is in calculation for the ventral paraformation. There is a habitual condition that happens. So you can see that molecule is here labeled in green. It is present. You can see the appraisal is in the body itself. There is no molecule. It's here where it accumulates to constrict the apical area. And then if you view it from the top, this region will be lower in area than this region. So these regions here are smaller area compared to these regions here which are large and invasive. So and then this is in neurodevelopment is the actual second monopod formation. So here again you have you see the myosin which is exists here and not present anymore here. And the myosin which is specifically reduced here. So basically is expanded. So there is much less myosin. Okay. So now this is one particular example that I given for tube formation. And the other one is so how do you elongate a tissue? Padma had told us there are two different ways in which you can do it. Either you are doing oriented cell division, in which way the width of the tissue is actually not really changing. You are just elongating it, that is adding more and more substance to the cell. In internalization, these cells go between each other, such that now the width of this tissue is reduced while also increasing the distance. So now we'll try to understand how in order for these things to happen, how do the cells make these things happen? So let's say you have let's color the cell. So here I have one row of these cells blue, whatever the color are, and then when we intercalate, you see these intercells and this intercell. Okay, and so now I want to ask, so how can I do this? If cells, if I consider them rigid, what is the problem? What is the problem in that we might face during intercalation? So let us say I cannot change the shape of the cell, and I'm just going to move it to intercalation. Then, so let's say I'm moving one cell, and then there's an extracellular space created from somewhere, and another cell comes to occupy the space, and then an extracellular space created somewhere. And if this is only one two cell movement, in the whole tissue you see, if the cells are rigid and they have to intercalate, they will end up making gaps and holes in the whole tissue. And we don't want that. We want the embryo to be very robust. So then what is biology's answer to this problem is that you so let us see, this is the so there is one cell, there is a random one. So what it does is first it tackles the problem step by step. So first it will reduce the junction between two cells. this will lead to a formation of what we call the 4-point vortex. And then these two cells make contact and then increase. So in this way, what happens is none of the extracellular phase is you don't make extracellular phases. You don't expose extracellular. Okay. So in tissue level, if you see this is what happens. So it's not as synchronous as this. Not all of them produce the constituent in this direction together. But if you see these junctions, which are shown here, okay, so they are stream, and then this becomes a shorter with lesser with tissue, and then as they resolve these junctions, okay, they are supposed to be as they resolve these junctions, it becomes into a longer tissue. So now then okay, so in under the microscope, how do we see it? So what do we, how do people come up with these theories? What do they see? So, okay, fine. Before that, so for you to shrink these junctions, we do the same thing as we do for apical constriction. You just put myosin and actin there, and then they all shrink, and then by expansion there is also in this there is also acid that polymerizes here to like increase the surface area of contact. Okay, and okay. So now what do we see in this embryo? So this is okay. So this is a tissue in so Parma shown you the wing and told that integration happens there. This is a different tissue. Okay, this is a tissue which also undergoes elongation to integration. Okay, now you can see how the different rows of cells behave. So I don't know if you notice very well that like there is no cell division happening there.
Maybe you can take one more example. And was it
So I'm trying to I'm trying. I forgot to. Wait a second. Yeah. So if you see these two, you can notice these two cells. I said, but what is that this junction space and these two cells coming from. Okay. So you can try and see if you're able to just concentrate on these two. Right? So yeah, so this is how the this tissue achieves elongation, and with respect to okay, how do we know that myosin is present there, and how do we know that myosin is only causing this shrinkage happen? Like just a simple rule to show you myosin distribution in the cell during intercalation. Okay. I think highly pixelated. Okay. So green is myosin, magenta is a cell boundary. So white will be there when the green and the magenta are there. So when the myosin localizes at the cell membrane, you will see white. So wherever it is white, it is myosin present at the boundary. So you can see, I think, here maybe the entire slide, I know that is the junction here. Okay. So here you usually see white particle and then it becomes a point product and then after that point, there is no myosin there anymore. Then I don't know if it's clear. Okay. But here in this same tissue if you see, in the same tissue, so you can see that all the myosin enrichment specifically is in this axis because only these junctions are thinking. So along the section, the contacts which are oriented this way have myosin. Difficult more than contacts which are oriented this way. So yeah, so in these two ways, so what I'm introducing now is to the fact that cells have to regulate and they acquire a shape and they acquire this shape through the internal cytoskeleton machinery. And then so what I introduced now was actin and myosin, but there are more rigid structures which actually give the cell an overall shape. So that is a microtubule and it's similar to the steel rods and all that you put as scaffold for building the house. It is similar to that. It doesn't really undergo massive rearrangements for smaller cell shapes to happen. But yeah, so this is about the right thing. Yeah, so that's what.
Great, thank you. And stop the live stream, take a break, and then because we'll be at your work, you can ask them what you will do.
Right.
Yeah, or your voice should go on that. Okay, yes. So there was some question, it says like, is it possible to design an organism or can an organism develop in such a way that it can emit gas? Somebody I think is saying a lot of sci-fi. Is it?
So to partially answer your question, in zebrafish, a kind of fish that widely used in studying developmental biologists, what happens for creating, I would say, equivalent of our nostril, what happens is that there is a single layer of tissue and there are some different kind of cells which push these cells so that a hole is created on that single layer of tissue to form a hole from where gas exchange happens. So it is equal to when I say a sheet is sort of making a hole in a sheet to by different layer of cells by poking it from beneath so that a hole is formed and the gas exchange can happen. This is something which has been very recently studied in zebrafish.
Okay. Another question is about what are the proteins present in cytoskeleton that are responsible for determining the cell shape and the type? The proteins present in cytoskeleton that decide the cell shape.
Was this before we reached the cytoskeleton part?
Perhaps yes. Perhaps yes.
So the two proteins we mentioned here was the actin and myosin. Actin is like you could think of it as a polymer. It has a smaller globular actin which come together to form a filamentous actin. So if you take the smallest unit there, it's like a globular actin. And myosin is also another protein so myosin is heavily seen as the one responsible for any sort of fourth generation in tissues. because it only has the ability to, in some ways, move filaments against each other, creating a tension in the cells. But there is, and then the other thing is again microtubule, which is also a polymer, and which has made up of alpha and beta tubulin, and also microtubule is different from actin in one way, that is that it is not just a cytoskeleton, but it also carries this cargoes to their right places when they have to position organisms.
For example, when you saw that there were these myosin which were only accumulated at the apical end, it has to be mobilized from the cell to the apical end. So these microtubules act as tracks over which there are certain proteins that take these microtubules and walk towards, or rather move towards the apical end and accumulate them there.
I think the person is adequately sensible. There is perhaps a bit uncommon request and difficult one. Is there anything in Hindi literature that you know of that somebody can refer to and understand the thing that you are doing, or if you just explain it, if you are aware?
We are not aware of it, but we can maybe go back and post some info on their question.
Okay, okay. All right. So for the person who asked this question, we'll try and post something on our channel or on our webpage, and hopefully we'll have something. But as you understand, this is all cutting edge research stuff, and nothing practically happens in Hindi. English is the language of science for the entire world. And so this is it. So please, the best fastest way will be you please pick up some English. Sorry to say, it's an online question.
They might not be able to find that exact terminology. The terminology will be there in English, but you can find some YouTube videos which explain the entire process in Hindi. That is available.
All right. Okay.
But I also feel that language is not a barrier for science. We are from an institute which comes from diverse backgrounds across the country, and people come from very small places where English is not their first language. And despite of that, everyone is doing very good work there. So I don't think language is
actually, so this brings to a very interesting point. Like amongst our speakers, Charvi is from southern part of India, Chennai. Lamia is from Mumbai, and Padma is from Orissa. So I hope you appreciate, everyone appreciates this fact that science is all about interest, and it is not at all governed by language or religion. So with this, thank you very much, all the speakers. So we'll stop the language thing. We'll take some tea break, and then please feel free to ask questions to the speakers.